So today we will going to discuss a new species of Clostridium, that is Clostridium tetani. Clostridium tetani. And Clostridium tetani is the causative agent of tetanus, and the disease that is now relatively very rare in well-developed countries. And this tetanus has been known from very early times and having been described by Hippocrates and Arrhenius. So, tetanus, we will discuss about this Clostridium tetani, and nowadays it is very rare disease in developing countries. So, even then we must have knowledge of this disease, and it happens rarely. Now, we will discuss the morphology of this Clostridium tetani. The bacilli is a gram-positive, number one, gram-positive. Okay, इसके Clostridium में जितने भी species हैं they all are gram-positive, slender, पतला पतला, and measuring 2 to 4, 2 से 4 into 0.5 मीटर इसका size होगा, and the ends will be rounded. ये जितने भी होंगे ये सब इस तरह के होंगे, रैकेट की तरह या ड्रम स्टेम अपीयरेंस भी कह सकते हैं अपन. The spores are spherical, locate terminally, and twice the diameter of vegetative cells, as I draw this picture. ये पूरा रॉड है और इसके ऊपर ये जस्ट डबल साइज का स्पोर होंगे. And the spores are spherical, located terminally, and twice the diameter of vegetative cell, and giving them typical drum stick appearance. This is the drum stick appearance, or in other words we can say the tennis racket. And the spores does not stain with gram stain and appears as a colorless round structure. They tend to be pleomorphic and sometimes filamentous. ओके, ये अपना साइज एंड शेप चेंज करते रहते हैं और फिलामेंटस दिखाई देंगे, एंड दे आर नॉन कैप्सुलेटेड एंड मोटाइल एंड पेरिट्रिचेस फ्लैजिला आर देयर ऑन their surfaces. So this is all about the morphology. It is very important for the exam point of view. So you must have knowledge of all these things. And now we will discuss about the cultural characteristics of this Clostridium tetani. And this is obligate anaerobic, number one, obligate anaerobe 1, and the optimum temperature for their growth is 37 degrees centigrade, and the optimum pH is 7.4, and they can grow well in RCM, that is Robertson cooked meat broth, and thioglycolate broth, nutrient agar, and blood agar. In RCM broth, growth occurs as turbidity and there is also some gas formation. And the meat is not digested but becomes black on prolonged incubation. On blood agar, the bacilli produce a swarming, means thin spreading film growth. जो हम करते हैं कल्चर, तो वहाँ पर स्वार्मिंग होगी, मतलब एक पतली सी थिन स्प्रेड फैली हुई फिल्म बन जाएगी, ठीक है. एंड व्हेन वी डू कल्चर ऑन द हॉर्स ब्लड एगर, द कॉलोनीज ऑफ दिस species, that is Clostridium tetani, are surrounded by a zone of alpha hemolysis, and which subsequently develops into beta hemolysis due to the production of oxygen level hemolysis lysine, and that is known as tetanolysin. Tetanolysin. We will discuss later on. So this is all about the cultural characteristics, and now we discuss about the biochemical reactions. Biochemical reactions. It is feeble proteolytic but no saccharolytic property, and it does not ferment any sugar. ओके, किसी भी शुगर का फर्मेंटेशन नहीं करता है. एंड जिलेटिन लिक्विफिकेशन occurs very weakly, and the coagulated serum is rendered more transparent and softened but not liquefied, and it is indole positive. When we see the methylene there or MR test positive, we found positive, and BP again negative. And hydrogen sulfide, that is H2S, is not formed. And nitrates are not reduced to nitrites, and a greenish fluorescence is produced on media containing neutral red, as we see in McConkey medium. Okay, this is all about the biochemical reactions, and I am going very fast because we have discussed in earlier species of this Clostridium, so the repetition and the same again and again. Resistance and viability, we will see about this Clostridium. Resistance or viability. The spores may be highly resistant to adverse conditions, but the degree of resistance varies from the strain. Most of the spores are killed by boiling for 10 to 15 minutes, but some resist boiling for up to 3 to 4 hours. However, the autoclave at 121 degrees centigrade for 20 minutes kills the spores of the most spores. And spores are liable to survive in soil for years and are resistant to most antiseptics. And they are killed by exposure to iodine, that is 1% aqueous solution, hydrogen peroxide 10 volumes, and glutaraldehyde 2% within a few hours. If we have a solution of 1% aqueous solution of iodine, 10 volumes of hydrogen peroxide and 2% ग्लूटारल्डिहाइड में अगर हम सॉल्यूशन में यूज करते हैं और इनको एक्सपोज करते हैं तो द स्पोर्स कैन बी किल्ड विद इन ए few hours. Then we are going to talk about the toxins. Yes, toxins. And this tetani produces two distinct toxins. One is heat level, oxygen level, hemolysin, that is tetanolysin. Tetanolysin. It is the heat level, oxygen level, okay, and it is known as tetanolysin. And second is tetanospasmin. Tetanospasmin, and it is a powerful plasmid and coated heat level neurotoxin. It is a neurotoxin, and this is one is hemolysin. Okay, these are the differences between two. So, and both are antigenically and pharmacologically distinct, but their action is different and their production is mutually independent. एक दूसरे से का कोई लिंक नहीं है. तो you must bear in mind that there are two types of tetanus toxins. One is tetanolysin, that is heat and oxygen level, hemolysin, and second is tetanospasmin, that is also a neurotoxin, and it's a powerful plasmid and coated heat level neurotoxins. So these are the tetanus toxins, and now we will discuss about the pathogenicity. Pathogenicity of this is the spores of Clostridium tetani are ubiquitous, means they are found everywhere in our environment, either soil, either water, either air. Okay? And they occur in the gastrointestinal tract of man and animals, and they are especially present in manured soil because they are present in gastrointestinal tracts of man and animals. They are little invasive power, and tetanus develops following the contamination of food with Clostridium tetani spores. अगर कहीं हमारे घाव लग जाता है, चोट लग जाती है, और वहाँ हम कोई भी आर्टिकल में अगर ये Clostridium tetani हो, तो we will be infected, and the source of infection may be the soil, dust, feces, etc. And infection strictly remain located in the wound, and the disease is due to the effect of a potent diffusible exotoxin, that is tetanospasmin, and the toxin exerts its effect on the spinal cord, the brain stem, peripheral nerves, and at the neuromuscular junction, and directly on the muscles. जिससे हम देख रहे होते हैं, इससे जो सफर पेशेंट होता है, लॉक जो हो जाता है, एकदम बिल्कुल अकड़ जाएगा, ठीक है ना, इस टाइप की जो चीज़ें दिखाई देंगी, एंड दिस इज ऑल ड्यू टू दिस न्यूरोटॉक्सिन tetanospasmin. So this is all about the pathogenicity of this Clostridium tetani. Now we will talk about the clinical findings. And this disease is characterized by a strong muscle spasm, that is spastic paralysis, and special clinical features including locked jaw और trismus. ये गर्दन की ये मसल्स अकड़ी होंगी और एकदम आपका मुंह टेढ़ा हो जाएगा. And it is all due to the rigid contraction of the jaw muscles, which prevents the mouth from opening. A characteristic grimace known as rhizus sardonicus. Rhizus sardonicus. This is the picture you will see in your books, and exaggerated reflexes are seen clinically. And the opisthonos is pronounced aching of the back due to spasm of strong extensor muscles of the back, and is often seen in patients who are suffering from trismus. And respiratory failure may ensue, and a high mortality rate is associated with this disease. And these are the clinical pictures you will show if the patient is suffering from tetani. Now we will go for the laboratory diagnosis. Laboratory diagnosis. Each species of bacilli or bacteria we will study under these headings from exam point of view and from knowledge point of view. The diagnosis of tetanus is made on the clinical ground because isolation of organism can occur in the absence of disease. And it is also possible to have a disease, but unable to isolate the organism. So, a laboratory test only help in confirmation of our clinical diagnosis. Then specimen will be the bone exudate or the tissues removed from the bone. These are the specimen or sample for the test. Then we go for microscopy, and microscopy is unreliable, and the demonstration of the typical drumstick bacilli in bones itself is not diagnostic of tetanus. They may be found. Then we talked about the culture, and while we talked in cultural characteristics, we have talked on culture and the toxigenicity test that is done especially in organism, and it is meant for the research purposes. So this is all about the Clostridium tetani. I hope you have enjoyed this video. So thank you.